Hello, everybody. Welcome to our weekly reading session. Everybody is welcome to join. Details are in the description down below. Today, we are hosting Matthew Adrian, and we'll be presenting his paper, Denoising Drug Discovery Data for Improved Absorption, Distribution, Metabolism, Excretion, and Toxicity Property Prediction. It's a wonderful paper, and please enjoy the discussion.
A little bit about us. We work at Merck, and we're in the modeling informatics group located in South San Francisco, California. And in this group, I'm Matthew Adrian. I'm a postbac researcher. Just a few years ago, I finished my undergrad at Berkeley, and I think this year I'm looking to start my PhD in machine learning for drug discovery research. I'm here with my coworkers, Yenzi and Alan, who are also in the modeling and informatics group in South San Francisco. We're excited to share our most recent paper published in JCIM on denoising drug discovery for improved ADME prediction. So if you're interested in looking into our work a bit further after the talk, feel free to check out our paper. So first, I wanted to give a bit of background on the drug discovery and development pipeline. So this starts with target identification, which is essentially identifying a modality such as a protein that you want to target with a small molecule, for example, that is associated with a specific disease, and ends with the manufacturing of the drug such that we can sell it into market. When we're creating these machine learning models to predict ADME properties, this is mainly in the earlier stages of drug discovery, which helps with decision-making, such as prioritizing compounds for synthesis and experimentation and as leads, and for property optimization in the lead optimization stage. But first, I guess we wanted to talk a little bit about what are ADME properties. So ADMET stands for absorption, metabolism, distribution, excretion, and toxicity. So essentially what these properties are, it's a wide array of properties. They're a wide array of properties that essentially describe how a drug interacts within different processes within a human. So these are experimentally derived properties such as solubility, lipophilicity, permeability, and much others. So being able to predict these properties enables more efficient drug discovery because instead of using time-consuming and expensive experiments, we can predict properties for a large amount of different drugs using our machine learning models. Additionally, understanding the ADMET profile of a drug in general reduces late-stage attrition, which is essentially when a drug fails in clinical trials, late down the drug discovery pipeline. Because the ADME data is experimentally derived, this comes with inherent experimental error that is contained within all of our ADME data sets. Some major sources of this error are procedure variance, such as between chemists or over time as the procedure changes and improves, calibration issues with instruments that we use to measure these properties. Sometimes there's chemical degradation or impurities with our starting materials that we're testing. And when we curate data from multiple sources, this causes some variances as well. One way that we're able to measure and reduce noise within data sets is to take multiple measurements. For example, on the right, you can see this figure where we compare multiple measurements within a single assay to get a rough estimate of how much noise is within the data set. However, when you have data sets of tens to hundreds of thousands of compounds, it becomes unfeasible to test each compound multiple times because of the high experimental cost and the time that it takes. This experimental error that's present in our data sets limits the performance of our ADME prediction models. So we wanted to build a computational denoising scheme in order to both identify noise within the data sets and improve our ADME model performance. So some existing work in this area, there actually isn't much existing denoising work that has been done in our domain. Majority of our denoising work has been on imaging, such as fixing image classification labels and improving image quality. There has been some work on denoising chemistry and biology data sets, but these have mostly been conducted on classification tasks such as diagnoses or retrosynthesis prediction. This is a little different from our task at hand because ADMET data are usually regression tasks because of the continuous nature of the data. So there has been some work on regression-based denoising in chemistry and biology data sets. However, the denoising schemes developed in this work have been shown to be actually detrimental to model performance. So we wanted to find a denoising scheme that would actually improve our models. So as we were designing our denoising scheme, we came up with this general workflow. So first we start off with a noisy ADME data set, and then we wanted to determine a noise proxy or noise detection metric to be able to identify noisy labels, and with that information, be able to change the data or the model to get improved predictions. So the two major design questions in our work was first, what's the best way to computationally identify noise via the use of a noise detection metric? And what is the best way to change the data, such as filtering out the data, correcting the data, or change the model, such as fully retraining a new model or fine-tuning the existing model, such that we get the best improved performance from denoising. In order to test this, we designed and tested all our models on public data sets for reproducibility. So these 4 different data sets were taken from different literature sources, and they're all experimentally derived ADME endpoints, such that they have unknown inherent noise within each data set. Because we don't know the amount of noise in each data set, we wanted to add random Gaussian noise, artificial noise to each of the public ADME data sets to test our denoising methods. Doing this is important because to determine the efficacy of the noise proxy, it's necessary to know the amount of noise that's in each data point, so we know that we're actually detecting data points that have high noise in them. And we can use the artificial noise to control the amount of noise that is within each data set so that we're able to test our denoising scheme on a wide array of different noise regions. So to control this, we had 2 different parameters in our artificial noise addition function, which was just a random Gaussian function. This is M, the magnitude of the noise, which was applied to the standard deviation port of the normal function, and the percentage, which is the percentage of the data set that we actually added noise to. By combining these 2 parameters exhaustively, we ended up with 9 different noise combinations that we could design our denoising scheme on. Finally, within each denoising scheme, we used this base model architecture called ChemProp to build our small molecule property prediction models. This uses a directed message passing neural network. And how this sort of works is it starts with a molecular smiles, which is a string representation of a molecule, and it abstracts it as a molecular graph. So in this, the atoms are nodes and the bonds are edges. And these initial atom and bond features are initialized using a cheminformatics toolkit called RDKit. This graph is then passed through several directed message passing steps, which uses bond level message passing. What's special about directed message passing is that the message that goes one way, for example, from 2 to 1 is distinct from the message that goes from node 1 to 2. So after these message passing steps, we're able to get our final atom representation and aggregate them into a molecular representation, which is fed through a feed-forward network to get our final property prediction. So our first task in developing our denoising schemes was to identify the best noise detection metric. We surveyed a variety of different noise detection metrics from literature. The first one we tried has been used less in literature, but has been used in some of the regression denoising that we've seen. This is training error, and this is just simply taking the absolute residual in the data set. The forgotten events metric was originally a classification metric, which we adapted to our regression tasks. So what we define this as, a forgotten event happens in a single epoch n when the training error of that epoch n is greater than the training error of the epoch that came before it. So we looked at this over all training epochs and summed the number of forgotten events for each molecule in the data set. Our last two were ensemble variance and split variance metrics, which are also commonly used in literature. What differs between these is the ensemble variance. We got the standard deviation of predictions with 4 different models, 2 with varying initial weights and 2 with varying validation sets, and a split variance, which was instead a split on the original data set. So we built 3 models on different non-overlapping splits of the training data set. So in order to determine which noise detection metric worked best to identify noise, we looked at how that metric was correlated with the amount of noise we added artificially. So the results that you see on the slide are from the specific noise combination where the magnitude is equal to 2 and the percentage of noise added is equal to 30% from the log-D data set. However, we see results that are consistent throughout all 4 of the data sets that we tested and every single noise combination. And what's evident from these results is that the training error noise detection metric is much more correlated to noise than all the other 3 noise detection metrics. Looking closer into the training error metric, what's interesting is that actually as we add more artificial noise into the data set, the training error noise detection metric does better at identifying which molecules have high noise. We identified the high noise molecules by looking at the top 10% of the data points, by ranking the data points and taking the top 10% based on training error. What we see is that at the lower noise ranges, there's a lot more misclassified points than at the higher noise ranges. So these misclassified points don't have high artificial noise, but they do have high training error. So we wanted to look into these misclassified points to determine if there's a way to further improve the training error noise detection metric. One thing we thought of is we could look into hard tasks for machine learning algorithms to predict in chemical property prediction data sets. Some examples of these are compounds in the data set that contain unique atom types that are not as represented within the data set, compounds with low structural similarity to the rest of the compounds in the data set, and activity cliffs. What an activity cliff is this happens when there's 2 compounds that have very similar structures chemically, but they have a very high difference in their labeled property. So what we wanted to look into are these hard classes, or are these hard tasks the reason why those compounds are getting misclassified when we use training error as a noise detection metric? We first looked into the compounds with unique atom types. And as you can see, there's not much misclassification with these unique atom types. They actually generally follow the trend of the rest of the molecules. So we think that the misclassified data points aren't due to compounds that have unique atom types. In addition, we looked at a clustering of a structural fingerprint and visualized it using PCA dimensionality reduction. So in this plot, the X's are the cluster centroid and the red dots are misclassified data points, where these data points don't have any artificial noise added, but they do have high training error. What we can see is that there's not really a visualizable trend with this misclassified data in their clusters. For example, some of these filtered data points are very far from the centroid. Some of them are very close to the centroid. And in addition, there aren't really any aggregation of these data points into one or two different clusters. So from this, we conclude that it doesn't seem like the misclassified data are very structurally similar as well. Finally, we looked into activity.
Just a question here about the misclassified data points, and they are compounds with unique atoms. And does this mean that the instance of misclassification will depend on the contents of the atoms within the compound? So what I wanted to know from you, what are these compounds that are going to create this sort of misclassification commonly? All these unique compounds, do they have like a pattern in sort of the chemistry classification, or?
Yeah, so I guess what we're trying to do with this analysis is to try to figure out if there was like a trend with the misclassified data points. So we actually don't know exactly why these misclassified data points are occurring.
All right.
But our first thought was that since we're using training error, these as like a way to identify noise, since there are known tasks that are difficult for machine learning algorithms to predict in chemical data sets, these might be the reasons why these compounds have such high training error, but they don't have any artificial noise added, which causes them to get misclassified by our noise detection metric.
Okay, all right.
Yeah. Does that answer your question?
Sure. I
Okay.
thought there would be like you went ahead to actually dig them down and classify them. I was curious, what are these atoms?
Oh, okay. Yeah. So this is, I guess this figure is separate from the unique atom types. So from this analysis, we concluded that like the misclassified data wasn't from compounds that included unique atom types. And now we wanted to see if that there was any structural similarity between the compounds that were misclassified. So the reason why we do clustering here is to cluster the atoms that have similar structures. So the clustering was based off of a fingerprint that measures structural similarity between the compounds. But what we see is that there isn't really a trend within this PCA plot with the misclassified data either.
That's all right, I think.
Yeah.
Mm.
Yeah, so I guess the next hard task that we wanted to look into is called activity cliffs. And the way that you identify these activity cliffs is through this procedure that we found in literature, where you calculate a structural activity landscape index for each compound within the data set. So from the equation that's shown on the slide, in our context where we have noise in the data set, this SOLI value is dependent on the noise in the data set because the activity labels or the property labels are dependent on the noise in the data set. So in our artificially noised data sets, we define two SOLI or activity cliff identification labels. There is a noisy AC and the clean AC. So we calculated the noisy AC based off of the SOLI value with activity labels that had artificial noise added. And if you think about it in a normal ADME data set that has inherent noise added to it, these are our observed SOLI values in the data sets that have noise. And the clean AC values are calculated on the original data sets. However, again, in a regular ADME data where we don't know the true label of each of the data points because they have inherent noise in them, this clean AC label is sort of intractable to us. We're not able to determine which compounds are actually like in this clean AC label region. What happens is when we look at the clean AC compounds, these are actually, in certain cases, in low noise and in this specific assay, apparent permeability, we're able to actually identify a lot of the misclassified data points, and it also doesn't identify a lot of the data points that actually are true high noised data points. So if we were able to get the clean AC values in data sets that had noise, activity cliffs might be the reason why some of this data is misclassified. However, when we look at the noisy AC compounds, which is what we would actually see in a noisy data set, the AC compounds with the noisy AC compounds sort of overlap with the clean AC compounds, so they do still identify some of these misclassified data points. However, in higher noise regions, this metric gets degraded as more noise is added to the data set. And it then identifies a lot of our high noise data, which we don't want to label as misclassified because they're actually, these data points actually do have high noise. So in essence, with this hard task analysis, we think that the misclassified data might be coming from activity cliffs, but there isn't really a good way to identify these to improve our training error metric. So after this analysis, we sort of went on with this training error metric to develop the rest of our denoising scheme. This part is on changing and altering the data and the model. So as I mentioned before, for this, we use the top 10% of the training error molecules to identify the noisy data. This is the analysis that I just presented before. And now our next step is to determine what's the best way to alter the data and the model. So this is like either filtering or correcting the data or retraining a new model or fine tuning. So next I'll go into the different denoising schemes that we tested throughout these experiments. So our first class of denoising scheme used a full retraining of a model. So the first one that we used was just a simple filter model, which identified the noise based on training error, filtered the top 10% training error out of the data set, and retrained a brand new model based off of this. We also tested a mean correction model where instead of filtering down the data, we corrected it by the average of the label in the original data set and the label in the label that was predicted on the training set by our base model. And then we retrained it. For the prediction correction model, it uses same sort of scheme as the mean correction model, except the corrections are done by simply imputing the predictions from the base model back into the data set and retraining. The next class of denoising scheme that we tested was sort of a transfer learning approach, where instead, so we still filter down the data set using the top 10% training error as in the filter model, but instead of retraining a brand new data set, we fine-tune the model. To give a little context about what the fine-tuning is in this case, so what we did was we first trained our original model on the full noisy data set and then used the final weights from this original model to initialize the weights of a new model called the fine-tuned model with the same architecture. We then allowed this fine-tuned model to train at a lower learning rate and lower epoch number on this filtered data set. Finally, we compared these denoising methods with different baselines so that we could get sort of a better context of the performance in these cases. So we tried, one of our baselines was the ground truth filter, which since we added artificial noise into all of our ADME data sets, we knew the actual top 10% compounds that had high noise. So we filtered out the known high noise compounds and retrained it similar to the filter model. We also used a random filter model as a baseline, where the noisy data points were just filtered out, identified randomly. So essentially we filtered out random data points within the data set and then retrained the model. So as I mentioned before, we had 9 different noise combinations. And when we were looking at our results, we identified 3 different noise ranges in which our denoising schemes performed somewhat empirically similar. So this was the low noise range, the medium noise range, and the high noise range. And in this analysis, we trained all these models with the artificial noise added specified by the noise range. And we tested our models on a held out test set without any artificial noise added to them. So now I'll go deeper into the results for each of the different noise regions. So first I wanted to present on the low noise denoising range. So for these figures that you're going to see in the next few slides, the y-axis corresponds to the average change in R squared, which this means is it's the change in R squared from the base model to the R squared seen from denoising. And this R squared is, this change in R squared is averaged across the 4 public data sets that we did experimentation on. So what we can see from these results is actually that when there's little noise in the data set, there's not much room for improvement. It seems like our models are pretty robust in this low noise scenario because the average R squared decrease that comes from noise in this low noise scenario is about 0.03. In addition, our baseline shows that like the upper limit that we can get, improvement that we can get back from denoising in these scenarios is about 0.01. So it makes sense that most of our denoising schemes in this low noise denoising range don't have much effect on the performance of our model. The medium noise denoising range is actually where we see the most model improvement. So in this range, actually all 4 of the denoising methods that we tested improved the predictions from the base model, where the fine-tuning method had slightly better predictions than the other 3 methods. These models are the fine-tuning method. It's actually interesting. When we look at the average R squared decrease that comes from noise in this medium noise denoising range. This decreases about 0.15. So the performance, we get back about 33% of the performance that we lose when we added noise to this data set. Additionally, these models are performing about on the same order as a ground truth baseline and perform much better than the random baseline. So we think that the improvements that come from our models in this noise region aren't due to randomness. Finally, we looked at the high noise denoising range. Just a caveat about these results. Typically, this high noise scenario is unlikely in our experimental assays. Typically, there isn't this much noise that is inherent within our ADME data sets, but nevertheless, we still wanted to try this extreme case in our experimentations. In this, it actually seems that most of the denoising models that we tested degrade the performance other than our fine-tuned method. So we think that the filter method, since it's trained on a smaller data set, since it just filters out the noisy data points, it overfits the model on still high noise data, which causes this large decrease in performance. For the correction models, since these rely on the base accuracy of our original model, these correction models get degraded because the original model predicts such poor predictions that it's essentially adding back noise within our data sets when we correct the top 10% training error molecules. We think the fine-tuning method is able to retain important information from its initial training, which allows it to have a baseline performance of as good as the original base model. So just a summary of our different denoising metrics. We think that the fine-tuning method is the best and most robust denoising scheme because so first off, in the low noise range, there's not much room for improvement, and we see that all of our denoising schemes actually don't really have a change in performance. In the medium noise range, where we see the most improvement, the fine-tuning method performs slightly better than the others. And in the high noise range, the fine-tune method has the same performance as the base model, whereas all of our other models degrade the performance. So from this, we're able to conclude that this fine-tune method, we think, is the best for denoising regression ADMET data. Because the performance of this method either improves the performance or the performance remains the same depending on the noise case. Additionally, because we're using fine-tuning, this method is less computationally expensive. So looking deeper into why the fine-tuning works, we think that because there's the weight transfer from the initial training, it's able to retain some of that context that it gets from the initial training on the full chemical space provided. And then the fine-tuning enhances the predictions on the cleaner data set, which gives predictions that are as good or better depending on the noise case compared to our original models. So now that we determined the best denoising scheme, we wanted to benchmark this denoising scheme on different data sets. The first data set that we wanted to benchmark on was the QM9 data set, because all the experiments that we've done before was on ADMET data that had inherent noise that was unknown. But these the quantum mechanical data sets are created synthetically through density functional theory calculations. And because of this, we can assume that they're free of noise. Additionally, this data set is much larger than the ADMET data sets that we found in the public domain. It's on the order of 130,000 molecules, whereas the others were under 10,000 molecules. So what benchmarking on this QM data allows us to understand, first, because these data sets are initially free of noise, we're able to understand exactly how much noise is within each data set and in each data point. And we're able to see the generalizability of this method into other domains on only artificial noise. So here are the results that we got from benchmarking on this QM9 data set. Here we compare the mean absolute error. So in this case, a lower mean absolute error is better. Here we only present results on the medium noise region, but we actually do see similar results as in our ADMET public experimentation, where in the low noise regions and the high noise regions, the fine tuning method doesn't really have, it has the same performance as our original baseline. But we do see performance improvement in the medium noise region. One thing we wanted to look at is also how the performance improvement changes at different data sizes. And what we see is up to a limit, as the data set size gets smaller, the performance actually improves. And we think that this is dependent on the difficulty of the task at hand. And we also think that there's improvements when we lower the training set size, because at a higher data set size, the models are more robust to noise. So because they're more robust to noise, there's less room for improvement from denoising. Finally, we looked at the performance of our model on 7 different ADMET endpoints from our internal data sets. One thing to note is that these data sets are also much larger than the experimental data sets from the public domain that we were testing. In addition, we are presenting here in the table also the mean absolute error. So a lower mean absolute error is better. Finally, the last thing to note about these results is that each of these data sets, in this case, we didn't add any artificial noise. So all of the performance improvements from denoising that you see in this table is only due to denoising the inherent experimental error within these data sets. So what we see from this table is that we were able to improve actually 6 out of 7 of the tasks from our internal data sets. The one task that we didn't improve had a statistically equivalent performance based on the mean absolute error and based off of the error bars that we calculated. The last thing that we noticed is that we typically see that our solubility assays do have the most inherent noise in them compared to the rest of the assays that we presented here on the slide. And these are actually the ones where we get the most improvement from. So now just to wrap up this talk, some key takeaways from this are that we found that in the regression setting, training error works best as a noise detection metric, whereas the other noise detection metrics that were commonly found in literature that have been used for classification tasks don't work on regression tasks. We found that filtering out the top 10% training error data and fine-tuning on the filter data set yields the best performance. And this performance is best in the medium noise ranges. And for our fine-tuning model, it has no change in the low and high noise ranges, whereas the other models that we tested degraded the performance in the high noise ranges. Finally, when we tested our model on real-world drug discovery data, we found that 6 out of 7 of our ADMET property prediction models improved from this denoising scheme. So some future work that we want to look into is to, instead of using a fixed 10% value for the filtering, maybe we could develop a method that would filter the data based off of the actual amount of noise in the data set because the inherent noise in each data set varies. So by developing a method that's able to vary the amount of noise that's filtered, we can better balance the losses that come from removing data points and the gains that come from removing noise within the data set. Another thing we can look at is a further investigation into the misclassified data points. We think that it could be because of activity cliffs, but we want to see if there's a better way to identify these data points such that we can improve the training error noise detection metric. Yeah, and thank you all for listening. Thank you for inviting me to do this talk. I really appreciate it, and I'm happy to take any questions from the audience.
Thank you, Adrian, for this wonderful talk. My first question is about, I've seen like the training error, I think TE is the one that performs, I could be wrong, that performs best in high noise sort of regimes. And is there a particular reason? Could be I missed it.
Yeah, so I guess maybe if it wasn't clear to begin with, all four of the denoising methods that we ended up testing used training error to identify the high noisy values. The differences between these metrics or between these models was actually in how we filtered out the noise and how we changed the model. So, so these three models like fully retrained a new model based off of either filtering or correction, but all of them used training error to identify the noise within the data set. Similarly, the fine-tuned method used training error as well.
Okay, so it looks like, was it the fine-tuning method when the model was fine-tuned and away from the ground truth filter, the fine-tuning, is it true that the fine-tuning method gives quite a good value of R?
Yeah. So we see that in the medium noise case, the fine-tuning method performs the best. And when we look at the R squared decrease that comes from the artificial noise that we add to this data set, it seems like the fine-tune method is recovering about 30% of the performance losses that come from noise. In addition, when we look at the ground truth filter, obviously the ground truth filter is going to have a higher performance because it's measuring the upper limit of the amount of performance gain that we can get back. But these two models are on about the same order.
Okay, yeah. And then also for someone who is starting out into drug discovery and machine learning, one would wonder what is the best representation or what can be said about the noise within drug discovery dataset? What is this noise? Anything that can be said about it? And like, yes, I know it. You have presented it, but it might remain sort of abstract. But in order to deconstruct that, what can be said about this noise as a representation of data?
Sorry, could you clarify what you mean by
that? What I'm saying is we have drug discovery data and we maybe have these compounds and the target proteins, I don't know, or molecules. And then one wonders what can be said about the noise in the context of drug discovery data. Like, what could be this noise look like?
Oh, okay.
Yes.
Yeah, so I guess uh like whenever we perform experiments, since all of this data is experimentally derived, we have to assume that there's noise that come from the experimentation. So the major sources from that create this noise within the data set comes from like procedural variances that come from different chemists, or possibly like the assay procedure might change over time. But the data set combines the results that come from all that come from each different procedure. So that creates noise within the data set. When we're measuring, there could be like different calibration issues in the instruments that we're using. Um, when we're looking at the starting material for some of our experiments, there could be some impurities within our starting material that causes, for example, in like solubility, the starting material not crystallizing properly. And finally, like when we combine data from multiple sources in the public domain, this inherently adds error within this data set because, again, they're coming from different procedures from different labs, different chemists doing the experiments, and errors such as that.
Okay, that's great. Then another thing, as machine learning, people want to hear about model architectures and and I saw that we are using the I think the deep message passing neural network, and
Yeah.
but also somewhere you talked about the Gaussian distribution where you're combining the ADMET dataset and I think noised data, but then I didn't really understand how the Gaussian distribution connects to I think the model itself. If you
Okay.
could throw some light there. Yes,
Yeah.
actually that's
So
light.
sorry. Yeah. Sorry if that wasn't clear. They're actually not related. So the Gaussian distribution here is just for adding artificial noise within each of the the public ADME data sets that we tested. Um, so here, the reason we're adding artificial noise into the data set is so that we know how much noise is contained within each data point. And also so that we can control the amount of noise that's contained within each data set so that we know when we're testing our denoising schemes, how they perform against different noise ranges. So that's how we derive the different noise ranges, is by adding this artificial noise using a Gaussian function.
Oh, okay, okay. That's
Um,
perfect.
yeah.
Then about the model, the DPMM here. Um, I see that we have this architecture in sort of a layered or tier sort of situation. And there's the first tier where we are seeing the deep MPNN, but also we saw the other models where we did the the tuning and the training on the TE. What's the connection here from this tier to the other one?
Yeah, so the connection with this and the rest of our denoising schemes is that I guess every time we built a model, we use it during using this architecture. For the fine tuning, for example, we would use this architecture to build our base model on the entire noisy data set, and then use the trained weights that are contained within like the message passing layers and the feed forward network to transfer this to a new model using this exact same architecture, and then fine tune that model on our cleaner filtered data.
Wow, that's sort of ingenious. Um, think I think those are these were my burning questions, and I would. Sorry, I have some paper around me, so But
Oh yeah, no worries.
those are some of the questions I had. I don't know whether there are members in the audience with more questions. If-
Yes,
I don't know.
yes, I have a
Dixon, you
question. Yes, I have a question.
are? Okay.
Thank you, Adrian, please, and your team for the presentation you've gave us in our group. I'm a PhD student in biostatistics, and I'm working on a Bayesian adaptive design in clinical trials. So I've liked your model, and I've been impressed. I want to know, this is the general question about the applicability of the methodology. Can this denoising model be applied to other phases of drug development beyond the preclinical data, such as efficacy and safety studies like phase 1, phase 2, and phase 3? Because you have a problem, 90% of the drug that has been developed into preclinical level fail. That's where you are. But from the 10% that succeed, almost 50 to 60% fail at phase 1, and still the death value of phase 2 drug still fails. So I'm suggesting, I'm asking, can this model be applied in the context of safety and efficacy studies?
Sorry, are you asking, I guess, can we use this model to predict whether compounds will fail in clinical trials?
Yes.
I'm not exactly sure that this kind of denoising scheme can be adapted towards that. I think for that kind of case, it might be better to I guess look at the chemistry of the compound and look at the ADME properties itself. And from that, we've actually seen having more optimal ADME properties was able to reduce the amount that compounds were failed in clinical trials. So I guess we can use these models to reduce the amount that compounds fail in clinical trials by optimizing for these ADME properties and learning about these ADME properties using these models, but it doesn't necessarily predict. We can't necessarily simply just use the machine learning model presented here to predict whether the compound will or won't fail. We have to more look deeply into the properties, and from that, like a chemist, for example, could use these properties to predict whether these drugs will fail in clinical trials.
But you can borrow some knowledge from your field, because being that you've used random assessment and clustering in your context, what I like about the drug development part of phase 1 and phase 2, safety and efficacy, now we are going into Bayesian model from the frequencies, which is the traditional way. So having anything like the model we're working on, I think it can be something very impressive when looked through that into details, please. Maybe in your future work, because we still have a bigger gap in this area. You can think about it. Thank you.
Yeah. Were you saying about using Bayesian models?
I said in efficacy and safety studies, that is phase 1 and we are mostly in drug development right now, we are adopting the Bayesian and adaptive way in the Bayesian. We are leaving out the traditional way of the frequency way, So your model, I think if you go through, you think about it, there has to be something that can be adopted from what you've presented to us today. Because there is a sort of gap, and now most of the pharmaceuticals and drug financing agencies are leaving out the clinical trial part because they fail at stage 3 when they are putting out of billions and billions of dollars. So you having any concept that you can bring into board that can be integrated into maybe Bayesian, because the Bayesian model is one of the best model that has been developed ever. Okay, it gives you a chance to adopt whichever way you have, and then you are able to come up with something that is new on board. So that's what it was just a compliment, not a question, but maybe you can continue to service research, please.
Yeah, thank you. That would be interesting to look into as well. Like the Bayesian stuff, I'm not completely familiar, but it would definitely be interesting to look into.
Thank you, Dixon, and thank you, Adrian. So Adrian, this is just about finding out if you've made the code and everything publicly available, such that maybe members can peruse through your code and gain more context if it is available.
Yeah, so I guess in our publication, we published a notebook that has a brief tutorial on how to use the fine tuning